And greetings. Welcome to the Evidence Reasons Academy. I'm responding to something that evolutionary biologist Grynch Lynchpin had to an exchange that happened about a month ago. I was very disappointed in his responses. It's like he totally ignored a lot of what I said, did not address any of the major points that I raised with the issue of protein complexity, and I'm just going to have to now start to be a little more specific. I told him I would start to try to get even some more specifics to address one of his questions. So I do want to thank him for taking time to listen to what I had to say. But I'm afraid, even though he is a professional at this, the response, it's like, these were evolutionary biology responses. These were not mathematical, probability-based, fact-based, chemistry-based, physics-based responses. That's exactly the problem, is he's appealing to evolutionary ideas, talking about evolution hypotheses. I was talking about chemistry, probability. And he didn't offer one thing about chemistry and probability regarding the protein orchard. So let's show what was being discussed here. He wanted to understand what my issues were with the so-called protein orchard. And I'd said, everyone agrees there's a protein orchard. That's not what's in question. And he's saying it's not a problem for evolutionary biology. See, that's the problem I have. There's nothing that's a problem for evolutionary biology, not chemistry, physics, or probability. I have an issue with that. In their eyes, it's problem free if you can make if it looks like parts have common descent. I said, let's assume common descent for the sake of argument. We all agree that there's a protein orchard. You say it's no problem because there's common descent. That's a non sequitur. Even if there were common descent, if you need miracles along the way, it's not a naturalistic process. Then you've got a problem if you're going to, okay, if you want to advertise as a supernaturalist, as a non-naturalistic process or something that's far outside of normative expectation, that's fine, but that's not being done. They're pretending this happens quite inevitably. And I have a major problem with that. And I devoted a whole hour 44-minute show. I showed something with nuclear import export, the eukaryotic protein import export, nuclear localization signals that was not addressed one iota in terms of probability and statistics, physics, chemistry, you name it. I have a serious issue with that I would devote that much time. Not one point was refuted when I started to talk about complexity, not for the topoisomerase, not for the nuclear import-export problem. And if you're just going to say I'm conflating this or that, and you don't address the probability, that's the problem. You're totally ignoring what is problematic. The questions I raised are serious questions that someone with kind of some sort of intuition about how likely an event is relative to normative expectation. And, I just don't know what to say beyond that. I spent an hour and 45 minutes, and then this is what he had to say. He said, thank you, Sal. I'll try to find the time for this today. And he says, to be clear, you talk about protein orchards and then you go 3 billion years forward and make an argument about extant cell complexity. This makes no sense. The one had nothing to do with the other. Sir, I beg to differ. What happens today is all you've got access to directly as far as the molecular level, unless you happen to have molecular fossils, which we really don't have 3 billion years ago. So if you could just, the problem I have is if you could just imagine whatever you want to make what we have today, that is not science. That's speculation. Okay? You can call it science if you want, but it's low quality science. It's certainly not to the level of electromagnetic theory that has experimental confirmation. So yes, the one does have one to do with the other if you're going to try to do science. If you want to just make up whatever you believe and you say it's no problem, I'm not going to deal with the statistics of where it is today, then just make assertions. That's not science. If you're not even going to make an attempt to describe the initial conditions and the processes that take that initial conditions to the final conditions, if you're not even going to try to appeal to first principles, physics, chemistry, probability theory, what do you got except just assertions? Did it? Oh, it happened. It happened is not a, it's a historical claim. It happened. Okay, I grant that it happened, but the question is, how probable is it in relative to normative, the mean expectation, what we call kind of a normative expectation. And if those terms are not familiar to you, you can look at you look at the normal distribution, you have an expectation of what will happen, and the things at the tail are unexpected. If it's like 20, 30, 40, 50, a bazillion sigmas from the mean, you don't really accept it as a legitimate hypothesis. That's the problem I have. Okay, so now let's see what else he has to say. And I have some, I will also present a little bit, excuse me, I'm going to present a little bit of some of the work, some of my work also on this topic, not directly. It deals with the collagen and it relates to another comment he's going to make. And I talk about topoisomerase, insulin receptors, and then collagen. And this is a very interesting thing, and I'm just going to read parts of it. But before I go here, this is actually, I'm reading this part of my paper because it responds to something he said. So let's go back to our discussion. He said, overall, Sal, it seems you're confusing many separate hypotheses and lines of reasoning. I'm sorry, sir. The only hypothesis is I'm invoking probability and statistics from an initial state. That's it. I'm really not interested in trying to leverage evolutionary theory as a premise. I'm sorry, I have to reject it. The quality of that science is so low, it just does not qualify, as what should be your starting premise. Okay, so let's go on here. He says, imagine that there are these phases of transition from geo to life. Oh boy, I'm sorry. I lost this. This is, okay. We can outline them as geo, bio, prebiotic, cellularization, biotic cells, eukaryotic cells, multicellular life, organismal life. These are all right. These are what you define it. I'm just saying you got to explain it. However you chop this up, I don't have to conflate it. The responsibility is on the people making claims. It's simple as that. I don't have to be all this. I just said this is improbable. I pointed to one specific point in eukaryotic evolution to say this is problematic. It doesn't mean, okay, but I don't know how, okay, maybe we're talking past each other. I gave an example of a eukaryotic transition. I did not conflate it with origin of life. If I said there's a transition between prokaryote and eukaryote, I identified a spot. It was in my video. I said it's problematic going from prokaryote to eukaryote. I don't know how can that be a conflation? I'm identifying certain things. I did, okay, so hopefully that clarifies it. You must choose a spot here and stick with that. No, I don't have to stick with that. I only have to give an example, and I say this is problematic for evolutionary theory. And then I don't have to stick with that one example or that spot. I can go to other spots. You want me to go to Origin of Life? We can go into those problems too. James Tour has done a great job of showing the problems. So when I highlighted topoisomerase, if you're going to assume that's in the first cell or something that's even modestly complex, viruses, there's some viruses that probably don't use topoisomerase, but is a virus life. We're talking about cellular life. So I don't know that it's that hard. Why do I have to go in GeoBio and Geo prebiotic cellular? It's still a problem. Okay. So topoisomerase, look at the topoisomerase. Tell me where you think that emerged. And tell me why you think it's not a problem for evolutionary theory. If you say, well, that's not part of evolutionary theory, okay, fine. Go to the eukaryotic, prokaryotic transition. I gave a textbook video that's shown to biology undergrads or whatever, and you didn't address it. Instead, you didn't address it. I'm sorry. You wanted a professional discussion. I would have expected you try to address it. You could have said, this doesn't work. You have a good point, Sal. I can't explain the complexity. And yet you're asserting it's no problem for evolution. You had an opportunity to say, this doesn't demonstrate a problem for evolution because here's the proof. And if you don't have it, just say so. I would respect your comments more if you did admit that it's problematic for evolutionary theory. And if you're saying it's not, you have to give a reason in terms of complexity, because the question was the problem of complexity and integrated function. So let's go back here. That would be a discussion for GeoBio and prebiotic cellularization. At this point, the massive number of proteins families is in place. Now it is not an orchard, but rather a network. Sir, that comes from Wolf and Kunin. They talk about the web of life, and may have been borrowing a term from what's his name, Doolittle. Do I have? I had a picture specifically where there was an orchard. It was so clear. You had collagen and you had zinc finger. Are you saying that's a network? He said in early prokaryotes there was splitting and asymmetrical splitting, budding and ingestion, horizontal gene transfer, and viruses. Are you saying the entire gene is transferred? Then yes, it might be a network organismally. But if you're taking piece of a protein here, piece of a protein here, stitching it together, that's very problematic. It may happen. But to have a coherent protein where you take a piece of one and put it in another, that's going to be, that just, the normal expectation is you're not going to have something functional. I'm going to explain some, when we go to specific examples, okay. All right, so let's say whether it's a normal expectation or not, for specific proteins, that just won't fly. You can't call it a network. And I've talked about zinc fingers on my channel. I'm just like, I don't know. I don't know how I can get you to try to respond and offer a probability argument. If you're going to keep asserting that the problem's mine, that I'm not understanding it, you have to explain it in terms of probability statistics from an initial state, the process that takes it from the initial state to the final state. If you don't know the initial state and you don't know the process that takes it to the final state, that is speculation. And how can you even claim that you even solved the probability if you don't even know what your starting point is? This is not science. We don't. the major law, the major theories of physics and chemistry are not built with that kind of speculation. It's fine if you want to believe it and you want, you want to speculate. Put it in the philosophy and religion department. Don't put it at the level of experimental science, please. Don't even put it in terms of something you figured out with probability and statistics to make assertions with. And if there's any conflation, it's happening in evolutionary biology, where they conflate common ancestry, evidence for common ancestry, with evidence that it solves the problem of complexity, the integration. Even Michael Behe, who accepts nominally common descent, he said that the issue is, how do we get all this integrated function? So, you see, you're turning it around and saying, it's horizontal gene transfer viruses, budding and ingestion, asymmetrical splitting. Sir, I gave you specific examples. Topoisomerase, nuclear import-export. Use everything you said and show that it is highly probable. If you're just saying, look at all these mechanisms and it's going to work, no, that doesn't cut it as a scientific theory. And you rightly point out here, however, what you really focus on is the complexity of extant life. That is in the latter organismal and late prokaryotic evolution phase of life. You conflate the extant with the ancient. Sir, we have complexity in the extant. If you're going to make claims about the past, you better have a description of what's there and justify how it goes from that state to the present. If you're going to say we have no clue, we don't have fossils, it's way back billions of years, then stop making any sort of pretense that you figured it out and that it's no problem. If you're going to assert that it's no problem for evolutionary biology and all you have is speculation, it's no problem because we have a speculation about the past that we have no clue about. That is not science. Does not solve the problem. That does not solve the problem. It only appeals to speculation, do not solve the problem. And you're saying, I can see how you're thinking about all this, and it's just plain wrong. No, sir, it's plain wrong to substitute speculation as fact. And the inferences have to be, okay, it's okay to have an inference if you don't have direct access to it, but the inferences have to be consistent with physics, chemistry, and probability. Probability within normative expectation. If it's not, it's okay to say it's way outside. But when you start to get way outside of expectation of first principles, you're no different than a creationist. You're invoking miracles without admitting it. It's actually worse than being a creationist because you pretend there's not a requirement to get to the details of that distribution, and when the theory would actually require it. And I will point out some of the challenges. He said, now I can grant you that the creation of single eukaryote cell by divine intervention. Okay, thank you. Now I can grant you, okay, for the sake of argument, the creation of a single cell eukaryotes by divine intervention 700 million years ago. Do you have the problem with the theory from that point to the present? And so this is where this discussion is going to go. Thank you for your comments. Because we can't interact directly, please forbear with me having to do something via video, as it enables me to use diagrams, and it is a little easier to at least simulate a conversation. Greetings, Mr. Jetty. Yes, the Grinch. Thank you for taking time to watch the video. I'll try to respond soon. And I'm sorry a month has gone by, but I have to gather parts of drafts of papers that I'm trying to publish to respond to your points. This is actually a paper that I was working on even before I met you, Grynch. This is a chance also for me to have an evolutionary biologist like you to review my claims before submitting to a journal. Thank you again. Okay, so he said, now I can grant you the creation of a single cell eukaryotes by divine intervention. Okay, thank you. Okay, only for the sake of argument. Do you have a problem with the theory from that point to the present? That's what he asks. And I'm going to show why I have a problem, at least with select proteins. So, I hope you were more than just granting it for the sake of argument. I hope there was something persuasive when I showed the nuclear import-export in a eukaryote. Going from prokaryote to a eukaryote that has a nuclear import-export complex is just huge. Now, granted, there are some prokaryotes, like this recent really big bacteria that has probably something similar to nuclear import-export, but even then, that's just another miracle. It's not really, I don't think people really believe that that thing was a step on the way to a eukaryote, that it's somehow ancestor or shares an immediate common ancestor with eukaryotes. But still, it's the evolution of complexity. You're correct that I focus on complexity. So let me show you my analysis of collagen. And you tell me, okay, collagen, all right, let's look at the Wikipedia article for collagen just so that I can make sure. All right. Okay, it says collagen, evolution of collagens. The extracellular matrix, oh, I'm sorry, let me show this. I'm sorry. I'm just grabbing this from evolution of collagens. It says here, the extracellular matrix is often defined as the substance that gives multicellular organisms, from plants to vertebrates, their structural integrity and is intimately involved in their development. Although the general functions of extracellular matrices are comparable, their compositions are quite distinct. One of the specific components of metazoan extracellular matrices is collagen, which is present in organisms ranging from sponges to humans. Okay, so this is metazoan. All right. Let's look at the term metazoan. So a metazoan, any animal that undergoes development from an embryo stage with 2 or 3 tissue layers, namely ectoderm, endoderm, and sometimes mesoderm. Okay, so it has to be an animal, and it undergoes development from an embryo stage with 2 or 3 tissue layers. Okay, so metazoan. So collagen emerged in the metazoan. So this is presumably then after the emergence of eukaryotes. So you asked, now I grant you the creation of single-cell eukaryotes by divine intervention 700 million years ago. Do you have a problem with the theory from that point to the present? Yes, I do, with select proteins, and I found one, and there are probably more that we could find, but I chose collagen for not in response specifically to you when I was writing this paper. It had to do with an evolutionary mechanism for making certain classes of multimeric proteins. Collagen complexes are multimeric. This one is a heterotrimeric protein that I'm looking at. So let's just look at human collagen, their principles that can be extrapolated to other collagens in other animals. So first thing I'm going to say, this was addressing the issue of frame shift. Okay. All right, so let me just read this. It's not, remember, this is directed at another evolutionary mechanism that is saying frame shifts can create proteins, and we were challenging, the limits of what proteins it can make. So I chose collagen as one example that says frame shifts won't make something like this. But as I describe why it's going to fail, why frameshifts will fail to make this, it will also show other mechanisms of evolution ought to fail too. So it says here, to the authors, so it says here, this is authors, meaning me and my co-authors. To the author's knowledge, and this is a draft, by the way, no one is suggesting collagen 1A1 or its ancestors arose via frameshift. In fact, it was for the reason collagen 1A1 is not proposed to originate via frameshift. It was chosen as a model of constraints that would preclude frameshifts as the mechanism of its origin or the origin of its ancestors. Frameshift mutations on human collagen 1A1 illustrates how frameshifts will cause the insertion of numerous stop codons and destroy its function by changing critical features. Frameshifts or anything that is highly randomizing will introduce stop codons. That's going to be very problematic. So random point mutations on non-genic space, that supposedly turn into a gene, it's going to be problematic. Especially if it's large, you'll have lots of stop codons and destroy it very quickly. And this is modestly long, about 1,000 residue protein. Okay, frameshifts on mutations illustrate, numerous stop codons. It cause the insertion of numerous stop codons and destroy the function by changing critical features. Examination of collagen 1A1 also suggests the degree of fortuity necessary to frameshift an arbitrary string to make a functional collagen 1 or its homologs or possible ancestor. Okay, so you can kind of visually see it. There's a pattern of glycines here. The G, every third residue for a very long stretch. It's a powerful visual pattern there. Okay, I mean that's just the tip of the iceberg. Collagen 1A1 illustrates the level of integration needed for the protein to be functional due to the many chemical partners required for its maturing. Collagen 1A1 forms a heterotrimeric protein that imposes geometric constraints on its shape to enable its polypeptides to partner with copies of itself and another polypeptide. Collagen is a well-studied protein and is also interesting because of its ubiquity in vertebrates. In humans, varieties of collagen constitute around 30% of human protein mass by weight. Functional collagen type 1 in humans is a multimeric protein composed of 3 polypeptide chains, 2 alpha-1 chains and 2 alpha-2 chains. Taking the faster representation of human collagen 1A1 from uniprot base and highlighting the glycine G residues, a striking pattern is readily apparent. And here you have it. You see the glycines are highlighted there. It's about 1,000 amino acids. And by the way, you have glycine can be implemented 4 ways. GGA from DNA, where the lowercase g is a, let me try to make this bigger. I'm sorry. The lowercase g is the DNA. The capital G is the amino acid glycine. So GGA, GGC, GGG, and GGT are the possible ways to make glycine, and you could see the different spellings there. The repeating pattern is serendipitous as it provides a means of visually conceptualizing a salient aspect of collagen whereby a glycine residue is present every third position. If there were no bias towards some sort of tandem repeating mutations, a random distribution of amino acids would not lead to this pattern. This is akin to the proverbial case of flipping a fair coin 1,000 times and getting heads each time. It would be a violation of the law of large numbers. So a frame shift, we wouldn't expect to do this. Maybe a copy mechanism, okay? It is known there is some tendency, so I'm going to grant this. It is known that there is some tendency for a variety of tandem repeats or copy variations to emerge. Superficially, one might postulate that the repeating glycine pattern is the result of a mutation in mechanisms that causes repeats. Since there are 4 glycine codons of 64 codons in the genetic code, under the assumption of equiprobable emergence of glycine, the odds of glycine appearing is roughly 1 in 16. Under the assumption of equiprobability, the odds of such glycine pattern in collagen 1A happening under this assumed distribution is therefore 1 out of 16 to the 337th power, or roughly 1 out of 10 to the 406th power, which would be a violation of the law of large numbers for this distribution. And I'll note, if you have a copy mechanism, the intervening amino acids are different. And it's even more subtle than that, and I'll explain later. It's going to be more difficult. This is not an exact copy. So I don't know that we have a mechanism that's going to make it every third. Not to mention, it's not the amino acids, it's the DNA that has to be copied here. So I don't know. Anyway, this is still I haven't gotten to the meat of the problem yet. Even this still should give one pause. Hence some sort of mechanism generating tandem repetition and or copy number variation would superficially seem an explanation for the pattern. However, tandem repetition alone cannot account for the repeating pattern, since the intervening residues between the glycines do not repeat, exactly what I just said. And the codons for the glycines are not identical either, just what I said. Hence, the origin of pattern may still be an open question. Okay, we just can't say, oh, it looks like it's just a repeating mechanism and it's solved. There's a probability problem here. All right. So now, using the XPASI tool to view the DNA sequence of collagen 1A, we examine the resulting translations. And though it could be argued stop codon frequency is below the expected level, any semblance of the same translated residue appearing every 3 coordinates is mostly gone in all the resulting shifted open frames, even though the nucleotide pattern GG is repeated every 9 bases. Again, we were specifically addressing what would happen if you have some random string being frame shifted. What's the likely it's going to make anything resembling something as complex as a collagen? And especially something that's violating the law of large numbers, that's not expected. So we can invoke a repeat copy mechanism, but it's going to fail if you combine it with a frame shift. Okay, so now we're getting to the meat. Beyond the repeating glycine pattern in collagen, the more significant factor precluding frame shifts as an explanation for the origin of collagen, okay, could be frame shifts. Anything else, actually, point mutations, insertions, deletions. Well, insertions, deletions can induce frame shifts, so they would have to, not change the reading frame. So that's a problem. So if you invoke insertions or deletions, you have one hurdle to overcome, or random mutation. So name it, whatever your mutational thing is. The more significant factor precluding frame shifts as an explanation for the origin of collagen is its multi-component features and integration into the context of an organism. Because collagen is well studied, many of its components are now publicly accessible through the UniProt knowledge base. The above diagram is now alternatively enhanced below to highlight the multi-component features. So this is one perspective of collagen, and I'm just going to take the same spelling but give it a different color coding so you could see things. Now I want you to these are the details that bother me about saying this just evolved. The color coding shows important features. When you see these P's here, these prolines, they will undergo a post-translational modification that's critical to the fold. It's way more complicated than just putting glycines every third residue. There are also, there's like a, this is a postal code. This is, I think, some sort of, it's like a localization signal. It's right here. It's a local, some sort of localization, I wouldn't call it nuclear localization, but it has to put it somewhere where it's a localization toward the endoplasmic reticulum. So, I mean, this is not trivial that you have to create a protein like this and then it has to go to the right location in the cell. There's so many compartments in a eukaryote. I mean, there are a variety of compartments in the eukaryote. It has to go to the right compartment or float around in some, outside compartments. But all this is a regulated process. I'm trying to show you just the requirement of the postal code alone right here. And then there are other parts of it, like this von Willebrand factor C domain that don't exactly know what that is. This part here in the gray will be excised off, and we're still figuring it out. So as it's maturing, you get pieces of the collagen cut out. So it will actually translate this from the mRNA, and then somewhere the polypeptide, as it matures, starts cutting pieces of itself off. And you need these peptidases and all sorts of excision type proteins that are specific to the collagen, or maybe a limited set of proteins, and then so it'll cut the header and then it'll cut the trailer here. So this NC1 domain will be removed and this header will be removed. It includes, the von Willebrand factor C and this whole excision region. How they figured this out experimentally is incredible. Okay, collagen requires important post-translational modifications of the P proline residues that must be irregularly interspersed between the regularly repeating glycine residues in order to achieve Col1A's biophysical properties. So these prolines will get a post-translational modification, and the ones that are post-translationally modified, they're about every 10 locations, amino acids. It's not exactly. And this, it helps it curve, but this is in the context of a multimeric system. It's not just folding this, but it's having some anticipation when it gets trimerized, heterotrimerized, it's going to have a certain fold. And it's like the paralog has to exist as well to make this work really well. It turns out there is a disease where if the paralog is not expressed and only the only one paralog, one of the paralogous genes is expressed, you get trimers that are homotrimers, not heterotrimers. That causes a disease. So, how did that evolve? If it worked well as a trimer, why then does it start to make why does the paralog start to vary and improve this situation? I mean, there have to be probability calculations for the selection pressure that can create that, and people just assume it exists. An assumption is not a demonstration from first principles that it's likely that a certain that a certain environment is likely to emerge. Just because it will be useful in some future environmental context doesn't mean it's going to be selected in the present environmental context. There has to be probabilities affixed to this. So let's continue. But this post-translational modification requires the existence of procollagen proline dioxygenase, also known as prolyl hydroxylase, that specifically targets the amino acid pair PG. Okay, so I need to point out you have this pair called PG, and that that's not highly improbable, but you don't want this prolyl hydroxylase. You don't want the prolyl hydroxylase just targeting anytime it sees PG anywhere in the cell. You really want it targeting stuff like collagens. So there's a problem in regulation, problem in targeting, and then it has to modify the prolines. The prolines have to be in the right position to enable these biophysical properties such that when it's post-translationally modified, it will create the correct fold. And I don't know how critical each individual one is, but if hypothetically it's non-existent, the collagen loses at some point. It will have effectively lost all of its properties. I mean, again, you'll say, well, it's just going to evolve it, but we know that if you don't have that prolyl hydroxylase there, what good is that? You need, okay, to have a selection pressure, if you need at least, when you need multiple components to be there, you have to assume then why did it select the individual components to begin with? If that's an assumption, that's not a proof of probability that it's going to emerge. Now, granted, we do have co-option, but still, this is problematic. Anyway, let me continue going with the architecture. Okay, it targets specifically the amino acid pair PG. Compromise of these post-translational modifications results in a variety of problems, such as scurvy. Okay, scurvy is very serious. You can't be having, okay, so maybe you're going to assume, okay, there was a creature that was using something, and so scurvy, it wouldn't be subject to scurvy. So let's say it's a primitive creature. Okay, let's grant that. But what's the likelihood prolyl hydroxylase is going to emerge at the same time your collagen did? If you're going to have at least two proteins to make a functional collagen, one, the collagen itself, and then the prolyl hydroxylase, this is really challenging to have two proteins simultaneously appear. And it's more than that. Collagen 1A also requires a sequence of amino acids known as a localization signal that provides a postal code that sends a raw collagen peptide to the right location in the cell. Then the mature collagen must be joined with a collagen 2 as another copy of the collagen 1 to make a functioning collagen. This is the heterotrimerization. And I skipped one point here. No, it's there. At some point, a substantial part of the raw polypeptide complex, known as procollagen, has its N-terminal and C-terminal ends cleaved off by an enzyme known as procollagen peptidase or proteinases. Further post-translational modifications, such as glycolization by disaccharide GLC, alpha 1 and 2, GAL, beta 1 and 0, on a previously post-translationally modified hydroxylysine, is conserved from sponges to mammals and important to function. I mean, okay, so procollagen peptidase, what was it? Prolyl hydroxylase, procollagen peptidase, proteinase, the collagen itself, some unnamed protein involved in the modification of the a glycolization of the disaccharide. that's at least 5 major requirements, and each of those is kind of like non-trivial. I mean, you don't get a random enzyme. I mean, even if any of this is co-opted, it has to be co-opted so that it can work really, really well with all this. You can't just, okay. The problem is I have is this has to be worked out. If they're going to hand wave and say it's solved, I'm going to hand wave and say it's not solved. This is not the way. I mean, it can be offered as a speculation, but it is not high quality science at that point. So let me show, see if I could find something on the internet for collagen maturation okay, collagen maturation. See if I could just find a diagram. I mean, this is painful to look at and to think that, okay, collagen synthesis, here you have it. So we have the mRNA, this terrible diagram, my apologies. See if they have a better one somewhere. That was a promising diagram and then they blew it because the quality is so bad. I'm looking for, I mean, this is so poor here. Let's see if I can, okay, here it is. Here's a better picture. So you get the translation from the ribosome in the endoplasmic reticulum. You have the hydroxylation of a pro and lysine. This is prolyl hydroxylase. Okay, then it's released and it goes, it gets this glycolization. That's a critical step. And then it's heterotrimerized. So I don't know if this is spontaneous, but you want to have 2 of the alpha 1 and then 1 of the alpha 2 and not, like all 3 alpha 2s. I don't know how the cell is able to segregate this spontaneously or mechanistically, where you get 2 of the one type 1 and 1 of the type 2. And then they're going to weave together and make a, a trimer. So this is it here, this triple helix. This is a heterotrimer. And this is still a procollagen. Then it has to be secreted from the cell. Oh my goodness, there is, you've got another localization issue. You have localization in the endoplasmic reticulum, some processing in the cell, and then it's secreted out. And we know that there has to be mechanisms associated with that. And once it gets there, the N and C terminal domains are removed, and then it can be stitched and cross-linked to other ones. To make a protein that can do all that, you can't just use phylogenetic diagrams and say it evolved, problem solved. I mean, the problem is it's one of you would need to have foresight to get it to work so well. This is why we don't expect a 747 to emerge out of a junkyard when a tornado passes through it. There have to be many coordinated steps in its manufacture, coordinated and compartmentalized steps. Because to have a working system, the parts have to connect and interface towards some working goal, and in this case it is structural improvement. And we're seeing how the molecule is built in steps to give it those structural properties. This seems really far from expectation if we have arbitrary DNA strings that are suddenly being turned on to become functioning genes. But I mean, there are so many requisite proteins to make a collagen system. Let's call it a collagen system. It's not just the collagen protein, but it is the system that makes a processed collagen as opposed to a raw collagen that is problematic. So this is yet another emergence of a protein that has an orchard model. It's a very distinct pattern, and that's why we can contrast it with any other protein. I picked this one specifically because it has such a visually compelling pattern, and you could see there's not any ancestral protein aside from another collagen and all these other requirements that have to be put on it. And this is long after the metazoan, long after the eukaryotic emergence. This is the metazoans now that are later in a different branch than, say, plants or any of the protists or whatever. Because this is now when you have multicellular. So this is in the multicellular metazoans. Yeah, because a metazoan has to have an embryo phase. So here's one that just had to, so even if you had a divine miracle for the eukaryotes, you have a little miracle for the collagen. And as I examined proteins that are well characterized, that are multimeric, Thornton may have published on one multimeric protein, but it doesn't necessarily extrapolate to others, like collagen. This is very, to me, this is extremely problematic, and I don't think this is, I don't think evolutionary theory is going to solve this, except by just assertion, and it doesn't solve the problem of complexity, and that's something that, independent of one's metaphysical views, this is unsettling to a lot of people. That it's like, well, you want to believe it, but let's not classify it as verified to the level of present-day operational theories like electromagnetism, celestial mechanics, geometric optics, quantum mechanics with in the field of chemistry. This is very speculative. So anyway, I'm just gonna close with just a song because I like having endings. Thank you for watching, everyone, and Mr. Jetty, thank you for visiting. So take care, and regards to everyone. And this is nice sunset music.